So, hi, David. It's Jennifer Chase, and I would like to first introduce you for a few minutes, and then you can launch into your talk.
Okay.
So, David Baker is a Nobel laureate, 2024 chemistry. He is the Henrietta Aubrey Davis Endowed Professor of Biochemistry at the University of Washington, where he directs the Institute for Protein Design and serves as an HHMI investigator. His lab develops AI and computational tools for protein design that have led to breakthroughs in medicine, technology, and sustainability. In addition, he has co-founded more than 20 biotech companies, translating these discoveries into real-world impact. Please join me in welcoming David Baker.
Right. Well, thanks for inviting me, and I'm sorry I'm not there in person. Can you see me okay and hear me? I'll assume that's a yes. So today I'm going to tell you about designing new protein functions using deep learning. And first, to put the problem in context, as you all know, the genes in our genomes encode proteins. They do that by encoding the amino acid sequence of each protein. And those proteins then fold up and to give rise to folded proteins, which carry out biological functions. So in a sense, the problem of biology is how to go from the genome sequence of an organism, like humans, through the proteins that are encoded to all the things that they do. And a first part of that problem is the structure prediction problem, going from sequence to structure. And so that's the problem of biology, and more specifically, the first part is the protein structure prediction problem. So what I'm going to talk about today is the inverse problem. So here, rather than working within biology and studying the proteins that exist in nature, we're going to be making up completely new proteins. And here we start with the function. It's really exactly in the opposite direction. So we start with a function that doesn't exist. For example, breaking down plastic or targeting pathogenic forms of tau for neurodegeneration. So we start off with that function. We hypothesize a protein or set of proteins that could have that function. We identify amino acid sequences which will fold up to such proteins. And then, since these are completely new proteins, we have to make them in the lab to see whether they actually do what they do. And so we work back to the amino acid sequence. And we know the amino acid sequence. We obtain a synthetic piece of DNA, a synthetic gene that encodes these brand new proteins. And then we take those synthetic pieces of DNA, we put them into bacteria or yeast cells or mammalian cells, who then produce the protein, and we can see whether it actually has the new function. And so today, I'm going to describe the AI-based design methods we've been developing, and then give you a number of examples of the use of these methods to design proteins across across medicine, engineering, and sustainability. And so the important point is we're always starting with a function that doesn't exist, and we're always ending with making a synthetic gene, making the protein, and then testing it. So what I'm going to show you is primarily, after I describe the methods, I'm going to be showing you the experimental results on the designed proteins. So the methods that we have developed are inspired by computer vision methods for generating images. Along the top, you see an illustration of DALL-E, the image generation program. So here, starting with images of all very large numbers of images off the internet, noising them to different extents, and then training a network to remove the noise. Once one's done that, one can start with a completely random image and then progressively denoise it And what one ends up with is a new image that looks like it could have been from the internet, like this picture of the cat here, but in fact, it's completely new. So we did exactly the same thing with proteins. We took all the protein structures that had been experimentally determined, we noised them to different extents, then we trained a network to remove the noise. And once we had done this, we found that we could start with completely random constellations of amino acid residues and progressively denoise and end up with a structure that looks like it is a real protein structure, but in fact, it's completely new. So that's what I've described so far as unconditional generation of images and unconditional generation of protein structures. Now, of course, unconditional generation is kind of a nice proof of concept, but it's not very useful. As I told you, what we really are interested in is designing proteins with new functions. And the way we do that is to condition the diffusion process on the function we want to make. So we're all familiar with conditioning in the context of image generation. You can say, generate an image of a cat sitting on a table. So I'm going to be showing throughout my talk different ways in which we can condition this protein generation process. So this first example is conditioning based on binding or based on a target. So we give it, in this case, the structure of the insulin receptor, and the diffusion process generates a protein in the context of the insulin receptor. And basically what's happening here is it's been trained to generate proteins around fixed targets where both what's being generated and what's fixed is from the protein structure data bank. So the program's learned what binding proteins look like. And so here, even though it doesn't have any particular example to go off of, it's generating a protein that looks like it should bind the insulin receptor. So what we do here is we then make a gene that encodes this new insulin receptor binding protein. And I'm not going to go into it in detail, but what we found is that this protein, in fact, antagonizes insulin signaling. If we put two copies together, we get very potent agonists, which have some interesting properties in that they work on even on patients that have insulin-resistant mutants, and they also don't induce. They don't have the cancer-causing side effects or cancer proliferation-inducing side effects of natural insulin. So it's turning out to be a very interesting molecule. So I just wanted to give you a few examples now of using this generative AI approach to generate proteins. And so here on the left, you see the TNF receptor. It's a major target for anti-inflammatory drugs. And here you see a diffusion trajectory that is generating a rather extended protein that makes extremely high affinity interactions with the receptor. It's in the single-digit picomolar range. We've been able to make versions of this protein that are absolutely stable in the gut, that cross-react with human and mouse targets, so we can test to see how well these work in animals. And in our studies thus far, this design protein is comparing favorably to some of the current drugs targeting this pathway. We can also condition the process on making particular types of folds. For example, we can specify that we want it to be an antibody fold or a VHH fold. And that is what you see here. We're generating, the diffusion process is generating a structure which binds the target, which is the influenza virus surface protein. And when we solve the cryo-EM structure, the actual structure is very close to the design model. For those of you familiar with antibodies, these are the CDR loops up here. So we can go beyond to design full antibody-like structures. And as many of you know, antibodies have 6 complementary determining regions, CDRs, and this compares the crystal structures with the design model over all 6 loops. So these methods can now accurately design antibody-like structures, which is neat. We can target other molecules relevant to the immune system. So our cells represent what's in their interiors using a system called the major histocompatibility complex, which presents peptides from internal proteins. So if you want to recognize a virus or infected cell or a cancer cell, which only differs based on its intracellular complement of proteins, these are very good targets. So this shows the diffusion trajectory that is generating contacts throughout the peptide, which is shown here. And we've made binders like this for quite a number of different peptide MHCs. And these can be quite specific for their intended targets and avoid off targets, as shown here for those of you who might remember a gene therapy trial a number of years ago, which went wrong because the molecule put in cross-reacted with a host protein titin. We can also design binders to G protein coupled receptors. We can design both antagonists and agonists. This is very exciting because G protein coupled receptors are some of the most important drug targets, and some of them have been very hard to specifically target with conventional approaches. So we can also now make binders to intrinsically disordered proteins. So these are proteins or peptides which don't have a defined structure. And these have been traditionally very difficult to target because they're flexible. There's nothing really to grab onto. But during the diffusion process, we can actually make this liability an advantage by allowing the disordered protein target, which is the dark colors, to fold up at the same time. We keep it sequence fixed because we know what sequence we want to target. We let that fold up at the same time that the binder is folding around it, as shown here. And so we've been able to make binders using this approach to quite a number of different targets. For example, amylin, the amyloid fibril forming protein. And I won't go into it in detail, but they have quite reasonable affinities and they bind their targets quite specifically in cells. So one class of intrinsically disordered protein that is which we're particularly interested in are the proteins that have been implicated in neurodegenerative disease, like A beta and tau. So we can make binders to these proteins or parts of these proteins. And what we find is that if you just take these proteins and put them into solution, they very rapidly aggregate. But if you add the binder, which is called a trap here, it completely suppresses aggregation and fiber formation. We can go beyond this by taking a binder, for example, to tau and fusing it to a component which targets the bound protein for degradation used through the proteasome system. And this is really neat because these are cells expressing tau. When we introduce this protein in, it completely eliminates the tau. And so we're now starting with collaborators to look at the effect of this in neurodegenerative disease models in mice. We're also making binders that are specific for phospho-tau, so we only degrade the disease-associated form. So now I want to switch gears and tell you a little about the design of protein nanomaterials. So for many years, we have been sort of fascinated by regular polyhedra and designed proteins that assemble to form cage-like structures. And my colleague Neil King developed the first de novo designed protein medicine, which was a COVID vaccine during the pandemic, based by placing parts of the RBD placing the RBD, the receptor binding domain of the coronavirus, on one of these cages. But one of the things we're very interested with this is biologics delivery. And the problem has been that in a regular polyhedron, you can't get larger than the dodecahedron, which has 12 pentagons. But if you're willing to break symmetry, we all know that you can get bigger than this by inserting hexagons in between the pentagons. And so we've figured out two ways to do this. One way is to use one type of protein subunit. And I should say these are two cryo-EM structures of designs that illustrate the two strategies we've taken to make larger cages. In this strategy, we use one type of subunit that has some flexibility, so it can alternatively be the edge of a pentagon or the edge of a hexagon. And you can see this comes out very precise. It looks just like a soccer ball. This is quite a bit bigger than current delivery vehicles. This is AAV for scale. And in this case, at each non-symmetry equivalent position, we've used a different protein chain. So here you can see what's surrounding the hexagons is different from surrounding the pentagons. And this strategy works as well. So now moving on to what we can do in the technology realm, we've been designing proteins that form channel-like structures. And when we insert these into membranes, we get conductance of ions across the membrane. Here you can see as we make the pore bigger, the conductance increases. We can turn these into sensors by designing proteins that bind to a small molecule and putting them at the top of the channel, as shown here, fusing them to the top of the channel, so that when the small molecule is present, the channel closes and no current goes through, as you can see here. So we're pretty excited about this as a very general way of making sensors for essentially molecular noses for what's in the environment. We can go further, and one of the things about protein design that is nice is you don't have to be constrained to what nature saw during evolution. And so for sensors, you might imagine it'd be more convenient and more effective to have them arrayed in silicon nitride chips rather than lipid membranes, because silicon nitride is much more robust. It's easier to couple to electronic readouts. And so now here I'm showing you diffusion trajectories here of a totally new type of pore that's the right size and the surface properties to insert into silicon. And you can see here that when we add these to silicon nitride, we have a high conductance, which comes down. We get a stable current. And so we're very excited now about using these for applications like protein sequencing. So everything I've told you about so far is design of proteins with single low energy states, but we can use these methods to make proteins that switch between states. So the idea is if we design a protein that when we add an effector molecule bends into a 90-degree bend like this, then if we build a structure out of this one, then when we add the effector, you can see if all of these things bend like this, the triangle will turn into a square. And for those of you familiar with biochemistry, this is allosteric because the action is far from the actual interface between these subunits. And this works very nicely. We chose not triangles and squares, not because there's a huge application, but it's very easy to visualize by electron microscopy. Here's the triangle protein, and we add the effector, and it turns into a square protein. Here's another case where we've designed a square protein to turn into a pentamer when we add the effector. Now that's kind of abstract, but we can use this concept to actually make useful things. So one of the problems with potent protein-based therapeutics is off-target toxicity. So you might want a way to turn a medicine off. And so we can use the same kind of action at a distance, allostery, to do this. So here we have a design protein that we've designed to bring together the IL-2 receptor subunits, the beta and the gamma receptor subunits, and this triggers a very strong immune response or inflammatory activation response. And then what we've also done is designed an effector molecule that when it binds, you can see it causes a conformational change and it breaks this interaction. And what this does is, if we look on cells, what happens when this effector is added, the two receptors, which are getting brought together by the drug in the first place, when you add the effector, they zoom apart. And what the downstream consequence of that is signaling shuts off very, very rapidly. So this is a general off switch for a medicine. We also can flip this around and make things that are on switches. So the medicine only turns on when it's in the right place in the body, for example, in the tumor microenvironment. Now, a recent exciting development is that we can now use these diffusion methods to design catalysts. So to do this, we start by just arraying in space protein side chain functional groups and sometimes cofactors that are in just the right geometry to catalyze a chemical reaction. And then we use the diffusion process to build up a protein around this active site. And this has been very exciting. So I haven't shown you much of the details about how we do these experiments, but in this case, we carried out this diffusion process 96 times. we encoded the results, the resulting proteins in 96 different synthetic genes, and we arrayed them in 96-well plate like this. And then we tested them for activity, and we found that the most active design had a very high activity. This is starting to approach the activities of naturally occurring hydrolases. I should say this is for breaking an ester bond. And we have been able to go beyond this recently with a series of bond making and bond breaking reactions. I'm not going to go into all the details. Two particular highlights is we can now break the peptide bond. And this is exciting because this is a very difficult bond to cleave. Proteins are stable for a very long time in aqueous buffer, so there's very little spontaneous hydrolysis. These designs break down are quite effective. They completely convert the starting protein to a product that has the target site, and they speed the rate of the reaction by 7 to 8 orders of magnitude, so 10 to 100 million fold. And this is very exciting because it gives a completely new way of thinking about therapeutics, because we can design therapeutics that not only bind their target, but also degrade it. So again, coming back to neurodegenerative disease, you could imagine proteins that not only block amyloid formation, but degrade amyloid once it's formed, or break down clots, for example. So we're excited about this as a new therapeutic modality. And then just to show you what the range of these methods are, we've been very excited about seeing if we can enhance the efficiency of photosynthesis. So photosynthesis in nature involves what are called photosystems, which have two chlorophyll molecules, which are positioned in a manner optimal for absorbing sunlight and using it for charge separation. And we can now design proteins that hold the special pair in exactly the same position. And we're starting to build these into larger light harvesting systems, as shown here. So here we've taken this special pair protein and assembled it into this cubic structure. The special pair protein is shown here. And now we're working to couple this with chemistry, where we can drive oxidation and reduction reactions with photon absorption. And then the final thing I wanted to tell you about is we can use these methods to make new types of materials. So, in our teeth are made out of calcium phosphate, and this is a very complicated process: bone, tooth, seashells. And what if we could design new materials like this? So here we've designed proteins that are complementary to a hydroxyapatite lattice, calcium phosphate, and we've designed oligomeric proteins which have the surface for driving these interactions inside. And if we take these proteins and we incubate them with calcium phosphate, we see the mineral forming inside. And then if we take these and we stack them to make tubes, what we see is that the mineral fills in the interior, and so we get these extended tubes. So we're very excited about being able to craft new types of designer materials that combine sort of proteins, which are organic, with inorganic substances. And a particularly exciting prospect is to use semiconductor materials. So here we have surfaces that are complementary to a zinc oxide lattice, and we find when we add zinc oxide that it fills in here. So this could give a new way of assembling semiconductor devices. And I wanted to say that we're also very interested in sort of improving the models that we use for doing these calculations. And we're doing this by trying to incorporate data from going well beyond the PDB, because as you know, for improving deep learning models, data is critical. I just want to show we released a general model for structure prediction called RF3 a month ago, and we've been able to improve it pretty much across the board by incorporating additional data, in this case, particularly data from metagenome sequences. So I think there's prospect for improving these sorts of models and then using them not only to infer to understand biology, but to design more and more complex systems of this sort I've shown you. So I hope you've seen today that we can use these AI methods to design new proteins for applications in medicine, technology, and sustainability. And I didn't talk about plastic degradation, but it's very similar to it's basically an ester bond breakdown. So we're using the methods I described to break down different types of plastics. And I just want to thank all the amazing people I've worked with, both in my lab and the general Rosetta community over the years. And thank you for your attention. Happy to take any questions.
Thank you. Thank you, David, for that brilliant talk. There are microphones here. So yeah, so why don't
I may ask a question from here. David, thank you for the talk. It's very exciting. When we work on diffusions in the context of computer vision to generate images, we use a text prompt to tell the diffusion what to generate. And we get an enormous diversity of solutions, all of which are compatible with the text prompt. And I was wondering whether you see the same in the space of proteins, where you get lots of solutions, and what is the rate, the hit or miss rate you get at the moment in terms of the function?
Yeah,
and then
it's a really good
whether
question. It's very similar. I mean, that's the beauty of these methods is they're really good at generating diversity. So we can we'll sometimes do as many as 100,000 different runs and get essentially diverse solutions each time. I mean, they'll fall into families, but to map out the space of possibilities, the hit rate is really a function of the problem. So if we're trying to, right now we're trying to make catalysts to break down forever chemicals, and there the hit rate is low. It's really hard for problems where the model has learned more, like designing protein-protein interactions. The hit rate can be quite high. So I would say it's very model dependent.
Thank you.
David, my question is that for your design protein for therapeutic purpose, have you, your lab, or your collaborator, or your startup company ever tested the issue about immunogenicity?
Yeah, so that's an important question. And so that definitely has been looked at. We've had a couple compounds that have been in human clinical trials, and their immunogenicity has not been a big problem. We think it's because the proteins are very stable and very soluble, so they don't get taken up very well by dendritic cells. But of course, it's an issue. We're also working on methods basically incorporating to the generative AI models understanding of for example, T cell epitopes, the MHC peptide presentation things. So we're now testing to see whether we can systematically increase or decrease the immunogenicity of designed proteins in such a way. Sorry, let me just close the blinds here, please. I should say sun in Seattle at the very end of October isn't usually a problem, unfortunately.
All right. Thank you very much for the talk. It was a very interesting deep dive into the current state of the art. I was wondering if you could give a bit more detail on like the practicalities and difficulties associated with going from a protein design outputted by your diffusion model to an actual physical protein. Like, presumably the diffusion model gives you secondary or tertiary structure. Do you then need to translate that back into primary structure and then produce a sequence like that? Is that like a difficult thing to do?
The model is the diffusion model I showed was diffusing at the level of amino acid residues. Our latest models were diffusing at the level of the atom. So we end up with an all atom structure. In some cases, we have a second model called protein MPNN, which will do a second round of sequence optimization. It's a graph neural network. So that part I would say is very straightforward. So, and there are tens of thousands of people around the world using these methods now to design proteins. We make all of our software available. I think the harder part is then when you want to go and make the protein in the lab, because then you obviously need a lab. And then the question is, how are you going to assay to see whether the protein does what you want it to do? And how many proteins can you make and how long does it take? I'm excited now about the prospect of doing closed loop sort of active learning sort of pipelines where you can actually go from a design protein on the computer to an experimental feedback within a day or two. But currently it takes longer than that.
All right, thank you.
Hi, David. Great talk. I just wanted to ask, These protein therapeutics you're designing are, of course, in very complex biological systems. And you mentioned off-target effects, but there's also things like the blood-brain barrier to consider. How much explicit guidance are you giving on all of these different sort of secondary metrics that matter for the therapeutics to your diffusion models as you're doing the generation? Or is it just
I think
sort of
that's a
implicit
good
from the
question.
data? Yeah.
Right, that's a good question. The answer is we put in all the information we have. I think one of the challenges in designing therapeutics is that there's a lot of biology we don't understand. So, for example, the question of what will get across the blood-brain barrier, our current approach is that we're designing proteins that bind to receptors, which are known to be at the blood-brain barrier. For example, for our tau degrading protein for neurodegenerative disease. But we can only put in what we understand. And I would say the real challenge as we move up the biological complexity ladder is that, we don't have as much detailed data on the higher order, physiological processes. So it's harder to put that sort of information in.
David, thanks for the great presentation. I have a very personal question. Can you elaborate the product that you're using, wearing the red sunglass? I assume this is for staring at the screen for a long time. I need to know the product name
That's right.
because I have the same problem. Thank you.
Oh, yes. Well, no, there's a lifesaver. I actually had sort of an eye injury a few years ago, and I couldn't look at screens, and it was terrible. And these just filter out blue light. They're called Cocoon. You can get them online really easily. They totally work.
Thanks, Dave.
Hi, David. I have a question, and it was really nice to hear you today. I want your suggestions regarding the docking models or docking tools for the ligand binding. So, we wanted to study more than one ligand binding. What will be the better suggestions, if you can say?
Yeah, well, there are, the best current methods for predicting the structures of protein small molecule complexes are programs like AlphaFold 3 from DeepMind. We have a kind of an open source version of method called RF3. And both of those are probably the state-of-the-art methods for, given a protein in a small molecule, how do you predict what is the structure of the complex?
Thank you.
Yeah, thank you, David. It's been always very inspiring, hearing your talks. My question is about data. So with the advancement of AlphaFold 3 and recent co-folding models, we're starting to saturate all of the coordinated information stored in PDB. What do you think could be the next step change in data? What do we need to do to acquire those data to make a next big improvement in this field?
Yeah, I think that is the central question right now, I think, for the field, for basically AI and biology, at least ground up biology. So I think there are two answers that I'm really excited about this. So there's certainly a lot of data that's distributed in the literature and that's been aggregated into databases. So there's public data, and there one just has to be sure to have some kind of data quality assurance. Then there's data that can be generated within a lab. And I think this is actually a great area for consortia. One of the reasons I'm really excited about using protein design for data collection is I told you we can generate, in answer to a previous question, we get many different solutions. And if we test things that a design model thinks should work, then they're kind of at the decision boundary in kind of an ML sense, because these are designs which the model predicts will work. So if they don't work, you get a lot of information from each such data point. So I think how to generate data as we go up the complexity scale will be really important. There's also privately held data, in pharma companies, and that's harder to access. But I think it's a tremendously important question, and I think some combination of public data, private data, and then experimentally, data generated particularly to improve a model, I think will be powerful.
Thank you.
Hello. Hi, it was a very interesting talk. So I have a question regarding the diffusing protein binders. So I just wanted to ask, does the diffusing protein binders increase the stability of the proteins or promotes its folding?
Yeah, that's right. So the diffusion process and the sequence design that goes along with it they're doing two things. They're first of all, they're ensuring that all the correct residues are in place to carry out the function, but they're also ensuring that the sequence encodes the structure. So both things are happening. And these, just to be clear, these proteins are getting made up completely from scratch. Many of them don't look like any proteins in nature. And so, yeah, so the sequence, the structure is encoded in the sequence that gets generated along with the function.
Also, sorry. So it also has an ability to restore its function. So, I mean, the diffusing protein binders also restore the function of the misfolded proteins. Is
Well,
it
there's
right?
no misfolded protein here. Here we're just making up completely new proteins on the
Okay.
computer.
Okay, yeah, got it. Thank you.
Statistics of the fraction of proteins you try to make in bacteria, yeast, or animal cells that are frustrated by cytotoxicity or degradation, and what are those numbers, and have you explored, for example, the use of coupled cell-free transcription translation systems in cases where that becomes a roadblock?
Yeah, it's a really good question. So when we test designs and they don't have the desired function, it's often hard to tell where the problem was. They could be perfectly functional, but they could have been cytotoxic. And indeed, we're switching more and more to cell-free in vitro transcription translation systems, also because it's faster. So when I mentioned we can go in a day from gene synthesis, gene product to or gene to protein to assay, it's faster. You don't have to worry about cytotoxicity. So we're doing this now for we're making proteins, for example, that bind to lipopolysaccharide, LPS. And you can't make them in bacteria. They seem to kill the bacterium, but you can make them cell-free. So yeah, so that's really for all the enzyme design stuff for different kinds of bond making and bond breaking reactions. We're really switching over to that. That's a good point.
Thank you.
Morning, David. As structural biologists, we're taught about the importance of ions and water in promoting the structures of proteins. And yet, and I've had the fortune of using some of your tools and software. And of course, the water is not explicitly carried out. Is this something that's just implicitly learned by the models? Or is this an area where we think these models can improve and learn more in time?
Yeah, well, I think that it's really primarily the first, that everything that you learned about the physical chemistry of proteins and protein folding is true. These models just basically learn it from training on 150,000 protein structures and protein complexes. So I think it certainly seems plausible that including bringing in some of the physical the physics into these models should improve things. So far, I would say the real problem with incorporating sort of physical principles is that we don't really know the parameters exactly. And I think the big advantage that the deep learning methods have had is there's a very straightforward way of obtaining the parameters by minimizing a loss by backpropagation. So the water is there implicitly, even though it's not. And now we sometimes do. So for example, one of the things I talked about briefly was metallohydrolases, where there's a water on a zinc. We found that we do much better if we explicitly model that zinc the ZNO interaction. It increases our yield of catalysts. So modeling in the detail explicitly can really help in the right places.
Hi, thank you for your talk. It was really interesting. I work with intrinsically disordered proteins, and I know you mentioned that you were able to train these models to fold the IDRs while it's folding this protein that you're designing. I was wondering if you tried doing this the other way, designing an IDR to bind to a specific ordered protein, and what are some challenges that you would encounter with IDRs specifically, given that they fold in multiple different conformations for a task like that?
Yeah, you're exactly right. So you're right. So I talked about designing proteins that bind to IDRs where the IDR is folding. So what you're suggesting is, well, what if we allow the binder itself to be flexible? And actually, we do that. So I didn't talk about today, but or I talked about it briefly. For the GPCR binder case, in some cases, the pockets are very recessed. And so, in fact, we just start with a sort of a peptide-like structure and let that be flexible, and it's sorting itself out when we make the designs. And my colleague at the IPD, Gaurav Bhardwaj, his group is really focused on designing peptides. But in the last step, we almost always try and make them more rigid. The reason is that you get higher affinity, or you in principle should get higher affinity if you make the disordered regions what's disordered rigid, because then you don't lose entropy of bond binding. One place, though, where we do use explicitly designed disordered proteins is for when I talked about the effector that was driving the conformational change. To drive that conformational change, we've found that folding upon binding can be very powerful. So it's almost like the power stroke of a motor. So I would say that's as we're thinking about designing motors and conformational changes, that's where we're really starting to think about explicitly designing disordered proteins.
Thank you.
Hi, dude. Thank you so much for your talk. I'm just curious. I'm a current undergrad in computer science. I really wanted to get more into biocomputation. I was wondering, do you have any advice for up-and-coming computer scientists to be effective collaborators and have an effective skill set to help biologists and in this kind of
Oh, well,
domain?
that's great. The field needs more people like you. I mean, I think really the best thing to do is to start collaborating with people doing this kind of work. You know, you can do that as an undergraduate. If you go to graduate school, you can do that. I mean, there's really no substitute for actually getting involved. As far as what you need, I think really the most important thing is just an interest in this area, because yeah, there's a huge amount to be done.
That sounds great. Thank you so much.
Hi, so my question is kind of where is the state of the tools these days in terms of a lot of the wet lab still does like antibody interference with receptors, and we don't know why. And I'm just wondering, with all the work that you're doing on modeling to construct new proteins to maybe bind, where are we now for being able to explain maybe autoimmune disorders and binding to dopamine receptors? So can we explain why they bind? And just where is the state of the tools at this point, and particularly your own?
Yeah, that's a good question. Well, I think the state currently is that there are really exciting ways to apply these methods to autoimmune disease. So, for example, if you have T cells which are attacking a particular tissue, one can make proteins which bind to those T cells and block them. And that's interesting because a number of autoimmune diseases, there's certain subtypes of T cell receptors which are prevalent. One can also target Tregs, and we're working on that now to try to amplify them in specific cases. So I would say the design side of this is the time right now to really try and advance autoimmunity, and it's something that we're working hard on, treatments for autoimmunity. I think explaining why the origins of autoimmunity is harder because it'll be weak off-target interactions of T cells for T cell receptors with some epitope they haven't seen. So I would say that in general, the modeling methods work better on strong interactions. I think as the methods improve, it should be possible to start identifying what those epitopes might be. But I think for right now, the best approaches are experimental sort of empirical approaches coupled with modeling for identifying the disease-causing epitopes rather than pure computation alone.
Well, thank you so much. It was really, if you have the antibody already identified and you have the receptor you think it's binding with, how can you in silico do modeling of why that's binding and what tools might do that?
Yeah, so if you know an antibody that binds to a target, the current tools can predict, in most cases, the structure of the antibody bound to the target.
Okay, this is the last question.
Yeah, my question is the current models, they model the data distribution. They have limited ability to generalize. So what are your thoughts on building more powerful models that can generalize better? For example, if some protein take nature 1 billion years to evolve, then the current method would be not be effective, right? Yeah.
Yeah, well, I would say the biggest surprise to me from all of the AI stuff that we and others have done on structure prediction design is that the models actually are pretty good at generalizing. The proteins that we generate and design, many of the ones I've shown you, don't have any close match to any structural match to any known protein. So, generalization is really a function of the size and richness of the data set and the ability of and the architecture of the model. And so one can generalize to quite a good extent. I think there are places where clearly more data is needed. For example, for protein small molecule interactions, the models are much less good than for protein-protein interactions because we don't have enough. So I think the key there will be, I think the most likely answer there is just more data. For example, design binders for many, many different small molecules. So I think, so I would say, I guess two-part, I think more data will definitely help with generalization. I'm not sure that different architectures, I'm not sure it's so much of an architecture question. I think it's more of a data question, but these models definitely, they generalize much more than I expected them to be able to.
Do you see a way to have the model continuously learn from more data, like in a more effective sort of flywheel?
Yeah, so I sort of showed that in one of my last slides. So distillation is very powerful for with these models. So you need, so for example, in the case I showed, we took several million metagenome sequences, generated models for those and trained on those, and that clearly improved performance across the board. I don't think totally free generation in the way that language models can currently do will be as powerful, but I think that remains to be explored. I mean, I think all the sorts of tricks that and approaches that are being used for the latest generations of LLMs, I think could in principle be applied here. So distillation, self-distillation, and whether one can do it with completely synthetic data or whether one always needs some experimental result still remains to be figured out.
Also, like reinforcement learning, like learning from like model proposed experiments.
Right. Well, that's, as I said, that's where we're really trying to go, to set up a lab in a loop thing so we can do experiments in line. And there, I think it'll be possible to learn very effectively.
Okay, thanks very much.
Okay, well, David, that was an amazing body of work that you talked about. And I think that all of us who are hoping for drug interventions for a host of diseases are thrilled that you're doing this work, besides those of us who just love chemistry. So let us thank David.
Well, thank you very much. And I'll just say we're really excited about collaborating and both with sort of AI developers and people with problems they want to solve. We have a particular interest in neurodegenerative disease now. So if any of you are interested in collaborations along any of these areas, just please let me know. And thanks again for your attention. I'm sorry I'm not there in person.
Thank you.